So the activity of pyridine receptors and dihydroxypyridine receptors is regulated by the following chemical, and the options are glutamate, calcium, magnesium, and sodium. So can anybody guess?
Calcium.
Okay, any more answers? Okay, so yes, the correct answer is calcium. And let's talk about these something about these receptors. So these receptors are basically calcium sensors. Okay, so both of these receptors are present on sarcoplasmic reticulum. Okay, and this is present in basically myocytes, and it is a storehouse of calcium. And it helps in maintaining the amount of calcium that a muscle cell needs for its signaling or for contraction. So it helps in storing that more than that amount of calcium, and whenever needed, it releases that particular amount of calcium for signaling. Okay. Now talking about these receptors. So these receptors are basically linked to each other. Okay. So whenever we talk about signaling of calcium, so the both of these receptors will come into play with respect to the muscle cells. So talking about the DHP receptor, it is the voltage sensing receptor. Okay. And ranodine receptor is the ligand sensing. So what happens whenever action potential comes to DHP? Okay. So it sends the signal to the ranodine receptor, which pushes it back, and then that helps in release or uptake of the calcium to the sarcoplasmic reticulum. Okay, so as can be seen in this case. So as the action potential reached to the DHP receptor, the R by R receptor is pushed back, and that helps in the release of the calcium. Okay, so this helps in basically whenever the muscle cell needs high amount of calcium, especially in case of contraction, when the myosin also needs calcium. So in that case, these receptors come into play and these release a large amount of calcium into the cells that helps in downstream signaling. Okay, so this is the major story of both of these receptors. So anybody has any doubt on this question? If not, then we'll move on to the next question. So the sliding filament theory explains the movement of the following molecules. So the options are actin and calcium, myosin and calcium, actin and myosin, calcium and acetylcholine. So can anybody guess?
Actin and myosin.
Okay. Any more answers? So yes,
C.
the correct answer is actin and myosin. So like what is sliding filament theory? It basically helps us to understand how a muscle contracts and relax on the basis of the stimulus. Okay, So this is the basic diagram of the sliding filament theory that I am going to explain. So these thread-like molecules that you are seeing are the actin molecules. Okay. And this structure that you are seeing is the myosin molecule. And myosin has the sites for binding of ATP because it whenever it moves, it requires ATP as the energy source. So it will have sites for all the three. Sorry, ATP, ADP, and PI. Okay. So what happens? First, the cross bridge between the actin and myosin is formed. So actin also has binding site for myosin and myosin has binding site for actin. So first what happens, a bridge is formed between actin and myosin. So next what happens, this bridge that is formed, so this basically slides before, a bit backward. Okay, this uh, leads to change in conformation of myosin chain. Okay, as explained in figure 2. Okay, now as this conformation changes, this leads to release of ADP and PI. Okay, and now what will happen when this ADP and PI are released? Then this there is conformation change in the myosin chain that leads to release of this cross bridge form. Okay, so now what will happen? The ATP molecule, a new ATP molecule will attach to myosin. Now the myosin will use this ATP to return like to change it into ADP and PI, and that will help myosin to bind to actin again. Okay, so this is the basic theory how a muscle contracts with the help of actin and myosin, and how myosin uses ATP, which helps in providing energy to myosin, which helps in binding to the actin, followed by the second cycle of contraction. Okay, so anybody has any doubt in this question? No, sir. Okay, let's move on to the next question. So the next question says cardiac cells exhibit a fill in the blanks action potential as compared to skeletal myotubes. So the options are shorter action potential, longer action potential, similar as compared to the skeletal myotube, or delayed rectifier. So can anybody guess?
Shorter.
Okay. Any more answers? So the correct answer is option B. And here is the explanation. Okay. So whenever we talk about the action potential in case of nerve, so what happens? The action potential has to pass very quickly, because if we are talking about a neuron, so consider this as one neuron, this is the axon, and this is the second neuron. So when we have to like pass the signal from one neuron to other, it has to be really fast. Okay. So in that case, the action potential will run very fast from through the axon, and that will lead to the next neuron. Okay. So in that case, the action potential in case of neuron will be the like shortest. Then we talk about the skeletal muscles. So in case of skeletal muscles, if the skeletal muscles are contracting, so in that case, they have to, like we just talked about the sliding filament theory. Okay. So I'm just taking an example to explain. So if the muscle cells are contracting, so talking of this as the actin and this as the myosin. So if like this has to be pulled backward. So when the muscle cells are contracting, so in that case, these has to be really fast. Okay, otherwise there is lot of damage to the muscle when we are uh, doing anything. Okay, so this is faster, like this is also fast as, but it is slow as compared to the nerve action potential. Now talking about the cardiac muscles action potentials. So what happens whenever we talk about heart? So we know about the flow that happens from the heart. So it has ventricles and atrium. Okay, sorry, atrium and ventricles. So consider this left, right. So now what will happen whenever blood flows through the heart? So during one cardiac cycle, blood enters the left atrium, then it goes to the left ventricle. Okay. So here are the walls that are present here. So these walls are open till the blood flows from left atrium to left ventricle. Now, whenever the blood is filled with the ventricle and left atrium is empty, then only these walls closed. Okay. Then the same thing happens with the left ventricle. So whenever it releases the blood, it contracts, and then what will happen? First contracts, then it relaxes. So during this point of time, the blood flows from the left ventricle to the arteries that are flowing after this. So it will wait till the blood is emptied into the artery. Okay, so that's why the action potential that we are talking about in case of heart takes much time to be followed. Okay, so that's why the action potential is very longer as compared to the skeletal muscle. Okay, so these are the same diagrams. Like I was explaining the concept, how can it be like measured in or compared in case of different systems, and this is the graph that we make in all the systems. So as we can see, in case of motor neurons, the action potentials take about 2 milliseconds. Okay, as I explained the concept. Then the in case of skeletal muscle, it's the 5 millisecond. Okay. And then in case of the ventricle, it's 200 millisecond, nearly 200 millisecond. Okay. So that's why this is the longest action potential as compared to the skeletal muscle or the neurons. Okay. So please tell me if anyone has any doubts in this.
So the shortest is in the neurons and next muscles.
Yes. So if this question is clear, then we'll move on to the next question. So dash cells are isolated from adult hind limbs of mammals to culture skeletal muscle. So the options are spindle, surrogate, seminiferous, or satellite. So can anybody answer?
Satellite.
Okay. Any more answers? So the correct answer is satellite. Okay. So what happens in case of muscle cells? So let's consider these satellites as a muscle stem cell. Okay. So we isolate these cells, and then what we do, we like these cells are present in a quiescent stage. Okay. So quiescent means they are growing, but they are not dividing. Okay. So they are particularly in dormant stage. So now what will happen? They'll form progenitor cells that give rise to different kinds of muscle cells. Now these progenitors will proliferate and give rise to myocytes. And when these differentiate, these will form long tubes called the myotubes. And like whenever there is some injury to the myotubes or injury to the muscle cells, then these cells that are muscle stem cells or the muscle satellite cells that we call, these are also present along with the muscle fibers. Okay, like these cells. So these helps in renewal of the damaged muscle cells. Okay, because these myotubes are already differentiated. But if these get damaged, they don't have the capacity to renew themselves. So these satellite cells helps in renewal of all the damaged muscle cells that is due to any wear, tear, or the age-related degeneration and something like that. Okay, so this was the case with the satellite muscle cells.
So are only
So,
satellites the muscles are used for culturing skeletal muscles?
sorry, can you repeat?
Are only satellite muscles used for culturing skeletal muscles or any other research also?
Yeah, so actually these, when we talk about muscle cells, so as I said, these myotubes that we are talking about, these are differentiated. Okay, so in case of vertebrates that we are talking about, these differentiated cells cannot be used for cell culture because in case of cell cultures, we need dividing cells. And if these cells are transformed in case of culture, then only we can culture them. So that's why in order to study the muscle scenario, we use the satellite cells.
Yes.
Okay. So any more doubts?
Are any other cells than satellite cells also used?
No.
Okay, sir.
Okay. So let's move on to the next question. So withdrawal of which of the following results in robust formation of myotubes in the skeletal muscle culture? So the options are glutamate, basic FGF, EGF, or acetylcholine. So can anybody answer?
Acetylcholine.
Acetylcholine. Okay. Any more answers? Hmm. Sorry, can you repeat?
ACFGF.
I couldn't understand.
Option B.
Okay, so the correct answer is basic FGF. Okay, so what happens in this case? We have fibroblasts also. Okay, so fibroblasts releases several factors. Okay, so uh, one of these factors is FGF. FGF means fibroblast growth factor that helps in growth of various cells, epithelial cells, or any other cells. So this is basically proliferating in nature. So it has two components, acidic FGF and basic FGF. Okay, so acidic FGF is has functions in the spinal cord, and basic FGF has functions in myocytes. So what happens whenever in case of muscle growth or muscle regeneration, first we need myocytes. So for proliferating myocytes, the basic FGF level increases. Okay, but when we have to differentiate, this is the case of proliferation. Okay, so now when we have to differentiate these myocytes into myotubes, so in that case, basic FGF levels needs to be down. Then only it will turn into myotubes. Okay, because this is the most differentiated. Now it will form the myofibers, leading to the formation of the muscle. Okay, so this is the basic scenario that we are talking about. So here, if we withdraw basic FGF, then there will be robust formation of myotubes. Okay, so if anybody has any doubt in this question, can ask.
No, sir.
FGF is fibroblast growth factor.
Yes. So if no more doubts, then we'll move on to the next question. So cardiac cells are connected to each other via sodium channels, sympathetic neurons, gap junction, or ciliary cells. Can anybody guess? So what happens in real scenario when we are talking about heart?
Gap
So
junction.
yes. So considering these as cardiac muscle cells, so these are very tightly bound to each other. Okay, because there are several factors. First, they have to contract and relax at different rate. Okay. And second, if there is and secondly, leakage of blood. Okay, so they needs to be very tightly bound to each other, all of these cells. So for that tight foundation, there are gap junctions present in every cell of the card, uh, cardiac muscle. Okay, so they are really tightly linked with each other and they have gap junctions more in number as compared to other tissues so that they'll be tightly linked to each other. Okay, so this is the reason why they have more number of gap junctions. So the cardiac cells are connected by gap junctions. Option C is correct. Okay, so anybody has any doubt in this question? So if not, then we'll move on to the next question. So dashed lines are classic characteristics of skeletal muscles, and the options are alpha, gamma, Z, and Q. Can anybody guess?
Z line.
Okay. Any more answers? Yes, the correct answer is D line. And here is the explanation. So this is the characteristic image of the muscle fiber that we are talking about. Okay. So it has different bands and different lines that are observed in case of the muscle. So A band, I band, M line, and Z line. These are the major uh, things that we talk about. So
Spare not visible.
so now is the screen visible?
All right, sir. Perfect.
Okay. So So I was talking about the structure of the skeletal muscle fiber that we are talking about. So the A band is called as the anisotropic band. Okay. And what happens here? Why it is called anisotropic? Because here both the bands, both the filaments are present. So actin and myosin, both filaments are present in this case. So that is why this is also called as the dark band. Okay. And considering this black line as the uh, actin filament and this yellow line as the myosin filament. Okay. So if we talk about the myosin and actin in both the places, then it will form a dark region in the muscle. So that is why the dark region is called as the anisotropic band. And in some regions, only actin is present, but myosin is not present. So in that case, that region is called as the isotropic band or the light. Okay. Now talking about the Z line and the M line. So Z line is basically the actin filament uh, region from starting to the end as shown in this image. So Z line starts from this region to this region, and M line is the myosin line. So where the actin filament doesn't touch or doesn't overlap with the myosin filament. So that is called as the M line. So this is the M region. Okay. So in the question, we are talking about the Z line. So Z line is the characteristic of the muscle size. Now, just some extra information about this. Whenever the muscle contracts, so what will happen? This actin filament move this side, and this actin filament from the left side move to the right. Okay, so what will happen? Both of these corners will move towards each other, and the M line that we are talking about decreases. Okay, so the same case happens with the, if we are talking about in case of the A band length decreases when the muscle contracts. Okay, and when it relaxes, it will be into the same position like the normal one. So this is the case of the uh, molecular level of the muscle cell, muscle fiber. And when we talk about the skeletal muscle, okay, so when we see the top view of the skeletal muscle, we will see some dark and light strips like this. Okay, this is because of these filaments only. Okay, so if anyone has any doubt in this, can ask. So if not, then we'll move on to the next question. So the joint of the individual skeletal myocytes to form myotubes is the process of phenotypic transformation, differentiation, second order division, or polyploidy. Can anybody answer? So as I have also previously discussed in some previous questions, so when we are talking about the muscle cell, so it's the myocyte. Okay. And whenever it has to do its function, it has to differentiate. Okay. So now it forms myotubes. So this is the process of differentiation. because it has been directed to a specific function. Okay, so it must be the process when individual skeletal myocytes forms myotubes. So this is the differentiation. Okay, so yes, this is the image I have shown you before also. So these are the myocytes that we are talking about, and these are the myotubes. So myocytes basically differentiates into myotubes. Okay, so if anybody has any doubt in this question, can ask. If not, then we'll move on to the next question. So dash is a common practice to obtain a reasonably pure population of embryonic motor neurons from embryonic day 14 rat embryos. So the options are immunopanning, centrifugation, enzymatic separation based on different activity of glial cells as compared to motor neurons, or the fourth option, application of ultra high centrifugal force. So can anybody guess?
Immunopanning.
Okay. Any more answers? So the correct answer is immunopanning. So what happens is, like, first when we isolate any tissue, okay, for example, considering this as the rat. Okay, so if we isolate, suppose any tissue, for example, if we isolate river, so in this case, first we have to do mechanical separation, followed by the enzymatic separation. Okay, this is the like non-specific way. And if we talk about liver, there will be hepatocytes and many other epithelial cells, blood cells, and many more type of cells that are present in case of the liver. Okay, so now if we have to be specific, so either we do ultracentrifuge that separates the cells on the basis of mass to volume ratio or the density of the cell. And this is not so specific because two cells, for example, if we take about the example of astrocytes and oligodendrocytes. So they have similar density as we discussed in our previous videos also. So we will not be able to separate both of these on the basis of the ultracentrifuge. So now what we have to do is we have to separate the cells on the basis of their markers that are present on their surface, because every cell has specific markers. Fine. So on the basis of the antibody staining of these markers, we separate the cells. So that's why this is the immunopanning technique. Okay, and this is specific cells, specific to a particular cell that we want to separate. So here we have to separate motor neurons. So we have some markers on the motor neurons also that we are going to separate. So we use those antibodies for against those markers and we separate those particular cells. Okay. So anybody has any doubt in this question? So if not, then we'll move on to the next question. So what is the role of side scatter in FACS, that is fluorescence activated cell sorting? So the options are: it determines the size of the cell, it determines the shape of the cell, determines granularity of the cell, or determines number of the cells. Can anybody guess?
Determines the granularity of the cell.
Okay. Any more answers?
Determines granularity of the cell.
So yes, the correct answer is it measures the granularity of the cell. And here is the basic principle of the FACS that we are talking about. Okay. So let's take an example of the simplest example that we are talking about, the motor neurons and interneuron. Okay. So for example, on the motor neurons, there is a receptor called A, and on interneurons, there is a receptor called B. So first, what we'll do, we'll stain the cells with their respective antibodies. Okay. So now we have stained the motor neurons with the antibody labeled as red. Okay. And for interneurons, we have stained the cells with the antibody labeled with blue. Okay. So now, because these cells will be mixed in a fraction, so we add these cells. These are differentially labeled cells. We add these cells to the sheet below. Okay. And now what will happen, because we have already labeled these cells with different antibodies. Now there will be some laser that will be pointed out to these cells while these flow through the instrument. So there will be two types of detectors present. First will be the front or the forward scatter. Okay. This forward scatter basically detects the size and shape of the cell. Okay, and then there will be other detector called the side scatter. This detects the granularity or the number of receptors or how complex the cell is being detected by the side scan. Okay, now what will happen? There is some like as this flows through the system, there are some droplets formed around the cells. Now these droplets will contain some kind of charge. So if we have to separate both of these cells, so for example, this will be given a positive charge and this will be given on the negative charge. So this depends upon the type of labeling we are doing to separate a particular cell. So now we will have deflector plates that will deflect different charge ion to different plates. So for example, if the charge is negative, it will be moved towards the positive plate, and if the charge is positive, that will be moved to the negative plate. So now we'll collect those cells, and these cells will be different, and these will be separated from a collection of cells that we were talking about initially. So now we can do any number of assays on these particular cells for their characterization or further analysis that we need. Okay, so this is the basic principle of fluorescence activated cell sorting. Okay, so if anybody has any doubt, can ask. So if no more questions, then we'll move on to the next question. So dash markers are commonly used for live cell sorting. So the options are nuclear, mitochondrial, R by R or DHPR cell surface or cell surface. Can anybody answer? So when we are talking about cell sorting, so for example, consider this as a cell, and this is the nucleus. These are the major organelles that are present here. This is the endoplasmic reticulum, or this is the Golgi body, and these are the centrosomes, ribosome, and there is the lysosome. So something like that. Okay, then this is the cytoskeleton that you are talking about. So now, if we see, if we separate any cell from fats or cell sorting, there are some receptors present on the cell surface. There are some receptors present on the mitochondria. There will be some receptors present on the nucleus and some on the sa- endoplasmic reticulum. Okay, so when because we need live cells and intact cells, so we do not do any kind of lysis or something in this case. Okay, so what we do is we stain these cell surface markers. Okay, based on different antibodies that we have. And these cell surface markers are the basic difference expressed. Okay, so we use cell surface markers only for separation of any kind of cells. Okay, not the organelle surface markers. So nuclear will be inside the cell, mitochondria will also be inside the cell, and these receptors are present on the sarcoplasmic reticulum that is also inside the cell. Okay, so we need cell surface markers to separate for live cell sorting. Okay, so anybody has any doubt in this case? If not, then we'll move on to the next question. So this is an analytical question. You cultured skeletal muscle cells and you checked the calcium levels using the BFPTA based fluorescence indicator. So this is a fluorescent indicator that helps to check the levels of calcium inside the cell in a culture plate. And we saw the increase in fluorescence in the growing cells. So what is your inference based on this? So the options are cells are attached to the surface, cells are growing properly, cells are growing properly and communicating with each other, and or all of the above. So can you guess?
All of the above.
Okay. Any more answers? So when we answer such analytical questions, we have to like just go to the basics that we study. Okay. So first thing is we are talking about the skeletal muscle cells. And secondly, we are talking about the calcium levels in these cells. Okay. So like I'll discuss later how we are using these BAPTA-based fluorescent markers. So first I'll explain the concept. So in case of skeletal muscle cells, we have myocytes culture and we need to study whether they are differentiating or not. Okay, just for example, I'm telling. So when these myocytes will be differentiated, these will be connecting or communicating with each other. Okay, then only they will transfer the signal from this myocyte to this to this, and the calcium level or the calcium uh, levels that they have will be high. Okay, so whenever we are talking about any functional assay relative to the uh, functions that they perform inside the human system or the in vivo system. So we talk about such assays. So if the myocyte is linked to other myocytes and is communicating with them, then the calcium levels in all the myocytes will be high. Okay. Now, if these myocytes are only attached to the surface and they are growing. Okay, for example, this is the culture plate and these are the myocytes that are present, but they are not communicating with each other. Okay, so in that case, what will happen? There is no signal from any of the myocytes communicated from other myocytes. So in that case, the levels of calcium will be down in this case. Okay, although they are growing in a culture plate, and they are attached properly, but they are not communicating with each other. So they will have low calcium levels in this case. And if they have high calcium levels, so that means they have attached growing properly and they are communicating well. Okay, so here the correct answer would be option C, because the calcium levels are not high uh, separated myocytes as compared to the when they are performing any specific function. Okay, so how we are detecting the calcium levels? We have used BAPTA-based fluorescent indicator. So BAPTA is basically this amino uh, aminopropyl tetraacetic acid based chelator. It is a chelator of calcium. Okay, so when it chelates the calcium ion, so what will happen? This tends to like change the spectra of the fluorescent marker linked to it. Okay, so that will change the fluorescence. For example, this is the dye that we are using, and it absorb the lambda 1. But when calcium is attached to this particular compound, so now the spectra changes and it will absorb to lambda 2. So now it will emit light based on the lambda 2 absorption. Okay, so this will help us to identify the fluorescence of emitted after absorbing lambda 2 and how much fluorescence it will uh, emit, we can also quantify. Okay, so all the calcium levels based indicator or sensors are based on this particular phenomena only. But the dye that they use will be different based on our need or our instrument sensitivity. Okay, so this is the calcium level. How do we check the calcium levels? And this is the basic functional assay that we do for checking whether the myocytes are functioning or not. The same assay can be used for cardiac muscle cells also. Okay, so the correct answer is option C. And please ask if there is any doubt in this question.
If the cells are growing, then it won't be attached to the surface. Is that meaning?
Okay, so what will happen when, like in the initial lecture, we also discussed whenever we grow cells in a culture plate, especially in case of adherent cells. So we see that whenever cells are attached to the surface, that means the cells are alive. Okay, and whenever they are floating on the media, that means they are dead. So here we are talking about the functional levels in case of calcium. uh, we have to check the levels of calcium inside the system. Okay, so here we are not talking about the live or dead cells. Here we are talking about whether they are functioning in a way that we want or not. Okay, so they can grow properly even when they are attached, or but they cannot have the high levels of calcium or the uh, threshold level of calcium when they communicate with each other. Am I clear? So any more doubts? So if not, then we'll move on to the next question. So why muscle cells is isolated at E18 from rats? So the options are to get some amount of satellite cells, to get minimal amount of fibroblast, to get maximum fibroblast in the culture, and or all of the above. Can anybody guess?
Option A.
Sorry, option A.
Option A, to get some amount of satellite cells.
Okay. Any more answers?
Sorry, sir, I correct myself, to get the minimal amount of fibroblasts.
Okay. Any more answers? So at E18. So E means embryonic and 18 means at the 18th day of the embryonic stage. Or here we can say that F18 also, because it's now fetus, not the embryo. Okay. So at this point of time, we isolate the muscle cells. Okay. So here we have two reasons for that. Because we can get satellite cells easily. And secondly, we will have minimal amount of fibroblasts. Okay. So here, both of the options can be correct. Either we can get some amount of satellite cells or minimal amount of fibroblasts. Okay, so here both of the options can be correct. Now, like first option is might be clear to you all because the satellite cells are the precursors for the myocyte. So we isolate these cells because these are the stem cells, as we have discussed before also. So it helps in easy culturing of the muscle cells. Now, why we need minimal amount of fibroblasts? Because as we talked before also, fibroblasts secrete certain factors. Okay, for example, FGF that we talked about. But here we have to grow myocytes first as compared to fibroblasts. So what will happen if there are fibroblasts, the number is more, that will outcompete with the muscle cells that we have. First point is this. And second thing is this, we have to like have more separation steps for separating these fibroblasts to get pure culture of the myocytes or the satellite cells that we are talking about. So we want the fibroblasts to be less in number when we especially grow muscle cells in case of in cell culture play. But here is one drawback also for this case for minimal amount of fibroblasts because these secrete certain growth factors like FGF that we were talking about. So what will happen when we culture with less amount of fibroblasts? So the growth of the muscle cells is quite slow in this case because we will have to add certain factors from outside as compared to the those secreted by the fibroblast. So that helps in more growth of the muscle cells as compared to when we add certain factors from outside. Okay. So here the correct option can be A or B both. Okay. So this is the reason why we isolate at E18 only. So anybody has any doubt in this case? So if not, then we'll move on to the next question. So adult myosin heavy chain is present in the options are differentiated myocytes, fetal myocytes, neonatal myocytes, or all of the above. Can anybody guess? So in case of myosin heavy chain, the literature suggests that when there is fetus, then we have the like post birth and then the adult. So in case of fetal, we have fetal myosin heavy chain. Okay. In case of post birth, that is the young or the child that we talk about, we have neonatal myosin heavy chain. And in case of the adult, we have adult myosin heavy chain. And here we have the differentiated myocytes. Okay, so the correct option is option A. So let's move on to the next question. So which of the following is a skeletal muscle cell line? Can anybody guess? So talking about C2C12, this is the skeletal muscle cell line that is isolated from mouse. Okay. And the rest three are cardiac muscle cell. Okay. So the correct answer is option 3, C2C12. Okay. So let's move on to the next question. So L15 media do not contain the following compound. The options are L-glutamine, sodium pyruvate, sodium bicarbonate, or all of the above. Can anybody answer?
Sodium pyruvate.
Hmm? Sorry,
Sodium pyruvate.
sodium pyruvate. Okay. Any more options? So here the correct answer is sodium bicarbonate. Okay. So like B24 media that we talked about for growing of neurons, uh, these L15 media is also generated to grow the cells in carbon dioxide free media. Okay, so it doesn't have sodium bicarbonate, but it has other salts like sodium pyruvate to maintain the metabolic level or the pH of the cells. So this is the case. So let's move on to the next question. Okay. So this was the last question for today. And if you guys have any doubt in the presentation or from anything in the week, can ask.
Sir, how questions are asked in the final exam? In the format of MCQs, or they can ask like theory or question answers like that?
Okay. Actually, as far as I know, they are asked in case of MCQ format only.
Sir, is there some book that you can recommend us to study from?
Actually, I also don't know about the book because we also refer to certain research papers only and the lectures that the sir only taught us. So that's why.
So, for how much marks they are asking exam? For 100 marks or 50?
Actually, I also don't have that answer because that depends upon course to course. And uh, you can ask this question in the discussion forum because the TAs of the course can answer that particular question.
So how often we do the cell muscle culture, and for exactly for what reason we do the cell skeletal muscle culture?
Okay. So the skeletal muscle culture is basically when we have to study the degeneration point of view from in cells. For example, if we have any diseased conditions, like take an example of muscular dystrophy. So either we have to go directly to in vivo systems like mouse or Labrador that we have, or the easiest system to generate is the in vitro system. So now if we have to test, suppose 100 molecules for that particular disease, we cannot go for in vivo system directly because there are two constraints. First is the side effect, and second is the cost. If we directly go to the in vivo system, the cost will be very large as compared to in vitro system. So first, what we'll do is we test those number of molecules in vitro first. So if any of these molecules will be behaving good in vitro, then only we can go in vivo. Okay. And secondly, if we have to study the progressive degeneration of muscle cells or any other diseased condition, and even in case of development biology also, when we have to study the development of the muscle, we cannot go to in vivo system. Okay, there also we have to isolate the muscle and then we have to culture it in our particular conditions that we are talking about. So different development conditions, then we have to culture those particular cells. So these three scenario I can now think of where the skeletal muscles can be.
Yeah, but then if you culture the cell skeletal muscles, probably it will multiply once or twice. That's all, and after that it will die. Is that the way it is?
Yeah, these are certain limitations with the cell culture system that we have, but this is the really cost effective scenario because talking about the funding about different projects, it's really less in academy. So for that, the cell culture system is the like very cost effective method to study all of these things.
So I'm not clear with that answer. My question was, if we culture the skeletal muscles, how long will it last for? That's my question.
Okay, so like talking about the satellite cells. Okay, so those are the stem cells that they can divide very much. Okay, now uh, we can maintain them as culture for as long as possible because these are the stem cells. Fine. Now, if you are talking about the skeletal muscle cells like differentiated myotubes, fine. So we cannot maintain them for so long. Okay, because those
It's a
are
clear.
yeah. So we will not be able to maintain the differentiated tubes or even the myocytes that are proliferated because they tend to differentiate into myotubes. So we'll not be able to like culture them for so long. But for these satellite cells, we can maintain that the culture for quite a number of times.
Clear, sir.
Any more doubts? So if no more doubts, then we'll close the session for today. And the next week will be the last session for the course. And I request if everyone has any doubts in the whole course, in any of the lectures or in any of the videos that are uploaded in the website, so can ask question next week. So for now, we'll be closing the session, and thank you guys for your patience.
Thank you, sir.